Thank you very much for the invitation to speak. Thank you everyone for coming out this evening, and I hope this is going to be interesting. I hope it's going to be useful. I guess we all come from slightly different backgrounds, and perhaps some of us are medics, some of us are not, some are from theological background or an ethicist background, but I hope this will be useful. Some of the medical bits that we talk about as we go through, I'll try and explain. For anyone who has come through medical school, paediatric metabolic disease is a very small print part of the curriculum, but it's what I do sort of full time. So hopefully as we go through, I'll explain some of that. We'll have the first slide, please, Trevor. So this is just a snippet from a poem, just to get us started, from Thomas Gray, who was an English poet from a few centuries back. And this is a poem, and this is just a few stanzas from that, highlighting how childhood should be in a real world, in a common sense, at a time when we are oblivious to the sufferings of life. Our happy hills are pleasing shade, our fields beloved in vain, where once my careless childhood strayed, a stranger yet to pain. To each his sufferings all are men, condemned alike to groan, the tender for another's pain, the unfeeling for his own. Yet why should they know their fate, since sorrow never comes too late, and happiness too swiftly flies? Thought would destroy their paradise, no more where ignorance is bliss, 'tis folly to be wise. This is a poem I came across, and he touches on the fact that all of us, all men, all mankind are condemned to groan. We all come across suffering in our lives. It's a universal experience. But in the innocence of childhood, where ignorance of our future sufferings is bliss, he says in that context, actually, to be wise to what will inevitably come to us all is in some way a bit of a folly. But if you come to the children's hospital, come to our metabolic ward or the clinic, or any other part of the hospital, and you are going to be confronted with the fact that often, tragically, children are not strangers to pain or suffering. Hi, come on in. There's something, if we think about the suffering of children and illnesses that they face, there's something instinctive in us that says this isn't right. There's something wrong if children are suffering. It's not the way it's supposed to be. And the problem of evil and suffering is a well-known conundrum, which in a nutshell asks, if there is a good and all-powerful deity, if there is a good God who is powerful, how or why does he allow such bad things to happen? And that question, I think, in my mind, is magnified when we talk about the suffering of what are potentially innocent children who have these horrendous diseases. How do we address that? And I wrote in the preamble to this lecture that any religious meta-narrative, any religious explanation that's seeking real-world credibility must at least attempt to provide an explanation for suffering. In other words, if we have a religion, if we believe that something holds some aspect of universal truth, it actually has to work in the real world. It has to be able to explain, or at least come to terms with, the reality of suffering. Now, the postmodernists, people like Jean-François Lyotard, have rejected to a large degree the idea of a meta-narrative, some big overarching truth, in favour of having your own truth, my truth, a localised or a personalised narrative. And that's the kind of the feeling of the day, isn't it? That everyone has their own truth. This is true for me, that's true for you, and there's a lot of relativism. And yet, if we have some seismic event that happens in our life, that really forces people to recalibrate that personal narrative. Suddenly the meaning of life has been shaken. And it's very interesting, and this is an observation that we see time and time again, that in that situation people will often go back to one of those traditional meta-narratives. They'll go back to the old religion. So in the hospital, often the chaplain gets called in by the family to tend to the family, to pray for them when they haven't set foot in church for years. So there is something about the kind of the old meta-narratives that are still holding foot. And what I'm going to try and do in this lecture this evening is, as Trevor said, a bit of an ambitious task, but to try and integrate the field of inherited genetic metabolic disorders and the kind of the latest things in genomic medicine and biotechnology with an orthodox biblical meta-narrative. In other words, trying to see how a biblical theology of genetic disease might work out. We move on, thank you. What I'm trying to see is, can the Christian religion, can the Christian faith, a biblical view, have something useful to say to some of the dilemmas that we face in the metabolic clinic? Being first and foremost a clinician, I want this to hopefully be useful and pragmatic and address some of the real questions and issues that come up. And I thought it'd be helpful if we crystallised our thinking around some of these questions. And these are the types of questions that will always come up when I'm speaking to parents in the clinic. The question always comes up: why has this happened? Then, once we know why it's happened, what can we do about it? Is there a treatment for this disorder that you've just told us about? And then thirdly, and particularly with these genetic disorders, the question is, what are the implications for future children? And I'd really want to just at this moment pause and acknowledge a lot of the parents and families that we deal with. I think anyone who's worked in paediatrics is always humbled by the way that parents cope with some really horrible things that are going on with their children. They teach us a lot of important lessons along the way. So first of all, why has this happened? When we're discussing a diagnosis of a rare metabolic disorder with a family, invariably this question comes up. And for our purposes today, the relatively easy part is the biology, the biological explanation, the how of why this condition has occurred. Discussing the diagnosis in terms of biochemistry, genetics, enzymes, metabolites, that bit we can explain. And I think clinicians are generally pretty good at that aspect of it. But where we're less good at ventilating is the more profound question, trying to understand why that has happened in the first place. So if the biology tells us how, we need to look elsewhere for the more fundamental questions of why has this happened. Now the biology, thank you, the biology is of the essence, and you do need to have some understanding of the biology before we can articulate some of those deeper questions. So if you put your seatbelts on, I'm going to give you a crash course 101 in paediatric metabolic medicine. So this is the simplified diagram. Biochemistry, metabolism. Just a quick show of hands. How many people in here have got some medical background of any sort? Just to get a feel. Okay, I'll try and make sure that everyone understands where we're going. Hi, good evening, join us, thank you. So all living organisms are complex, as you can see, complex biological systems, and there are thousands of different chemical reactions that are occurring every second in every cell of our bodies. And so the energy you need to power the molecular machinery that makes your muscles contract, or to fire a synapse between two nerves, needs energy. There are all these chemical reactions going on. You need chemical reactions to make new proteins that enable growth, and you have reactions that are recycling and degrading waste products. And our metabolism is the sum total of all of those reactions that's going on all the time. And every, or virtually every, chemical reaction that's going on is facilitated or catalyzed by an enzyme. And an enzyme is a specialized protein that helps that reaction to work. So thank you, next slide. So if we zoom in and we can keep pressing through this a little bit here, here's a simplified version. So chemical A gets converted to chemical B by our enzyme that does that process. The enzyme could be, you can think it a bit like a pair of chemical scissors. One of the things the enzyme does is chopping up these chemicals and changing them from one to the other. But if the enzyme doesn't work, we will end up with either too much of A building up because you can't break it down, or not enough of B because it can't be made, or A gets shunted to form something else that's toxic and bad for the body. If you've got that, you now have got metabolic disorders. That is inherited metabolic disorders by chemistry. Okay, now the protein that forms that specific enzyme, those scissors, is itself encoded by a unique gene. The nucleotide base sequence in the gene provides the blueprint for the amino acid sequence of the protein. To quote someone called Hubsvart, the genome is the primordial layer from where multiple circuits and complicated networks of molecular messages pervade living organisms. So you start with your genes, that gives you the instructions for the enzyme, and the enzyme then makes everything work. Now, if that enzyme doesn't work, that's because there's a problem in the gene. So if there's a misprint in the gene, if there's a mutation in the spelling in that sequence in the gene, the protein that's produced is faulty. It doesn't work. And because the enzyme doesn't work, you end up with blunt scissors or not enough scissors being produced, and that results in your metabolic disease. Is that making sense? Sort of. A few nods. So there are a number of different examples of these conditions. Some of them are relatively straightforward. You may have heard of the condition phenylketonuria, or PKU. That's one of the tests that we screen for on the newborn screening. So when a baby's born at a few days old, midwife will take a blood spot from that baby. That's testing for a number of conditions, but including PKU. And that's a condition where the body can't process phenylalanine. We can treat that very effectively with diet. But then there are other conditions, such as those related to glycine. Thank you, Trevor. Sorry, we're on here. Yeah. It's got a hard job keeping up with the slides. Glycine is one of the amino acids in the body. The amino acids are the building blocks of protein. Glycine is the simplest one of those, and it's got a number of roles. It's a neurotransmitter. It's involved in the communication between nerves, but you need it for building proteins and making other chemicals. Thank you, Trevor. And the main way the body gets rid of excessive glycine, if there's too much around, is something called the glycine cleavage system. And this is a whole series, a complex series of enzymes, a number of different scissors together that help break down the glycine to some smaller molecules, to ammonia and carbon dioxide. Thank you, Trevor. Now, I'm going to explain why I'm taking you through this in a moment, but just bear with me. I just want to show you how complex this process all is. So first of all, in step one, the H protein activates the P protein, and glycine is attached to vitamin B6 pyridoxal phosphate. Thank you. In step two, P protein decarboxylates glycine. That means it chops off a carbon dioxide and it attaches it to the H protein. And then in step three, the T protein uses some folic acid to remove an NH group, which makes the ammonia. And then in step four, the L protein reactivates the H protein, and we're ready to start going round again. And that's a simplification of the glycine cleavage system. But my purpose in perhaps baffling you with the details is to highlight what I call the exquisite complexity of our body's biochemistry. It's amazing when you study the biochemistry how intricate it is. It's an amazing system. But if this biochemistry goes wrong, if the glycine cleavage system doesn't work, you end up with a very severe disorder. And the condition that you get if this system doesn't work is something called NKH, non-ketotic hyperglycemia. You get very high glycine levels building up, and this is a problem that causes severe seizures. It even causes seizures to the baby before the baby is born. Women may talk about the baby hiccuping a lot in the womb because it's having seizures even before the baby's born. And it results in very significant developmental delay and often very early mortality. So this is one example of a very severe condition, and a lot of the metabolic conditions fall under that category. Often the bit of biochemistry that isn't working is so fundamental to our normal physiology that it's amazing that life can be sustained even for a short period of time when that bit is not working. Your biochemistry lesson is almost over. You're okay, but you're doing well so far. And the last bit we need to understand, though, is the inheritance or the genetic basis of these conditions. Now, the majority of the conditions that we deal with are what are called recessive disorders. Now, we all inherit 2 copies of each of our genes, one that you've inherited from your father and one that you inherit from your mother. And for a recessive condition, both copies of the gene for whichever condition we're looking at have to harbour a mutation or a fault to have the condition. If you've only got one copy of the gene that's affected, then you will be what's called a carrier for the condition, but you're not affected by it because the other healthy copy of the gene can make the enzyme, and so you have a backup supply. So in the usual situation, we see that both parents here are what are called unaffected heterozygotes. They're a carrier for one copy of the faulty gene, but the other copy is healthy. And then when they procreate, when a child is formed, you get varying combinations of those different genes. And the four possibilities are a child inherits both healthy copy, both wild type copy of the gene, and that child is healthy and unaffected, or they might inherit the dad's faulty copy and the mum's healthy copy, or the other way round, or in the unfortunate situation that they inherit both faulty copy, they then are affected by that condition. And we all carry mutations. It's part and parcel of being a human in the world as it is at the moment. A recent paper by Gao estimates that all of us have between 1 or 2 lethal mutations that we're a carrier for. And so if your partner happens to also carry a mutation in the same gene, that's when the risk of one of these disorders will occur. The combination of 2 carriers with a mutation in the same gene means that you have the risk of the disorder. And I mentioned here just the impact of consanguinity, marriage within families or cousins or other relatives. And that is a feature that we see in a number of populations. And that can increase the risk, because if there is a gene that's within the family, if you're marrying within the family, the probability of the gene coming together and causing the disease can be increased. And we see that, for example, with Tay-Sachs disease that's prevalent in certain Jewish communities. And some of those communities have taken proactive steps to screening, to seeing who is going to be an appropriate person to be my life partner, to try and reduce that risk. So the question then is, why do these mutations occur? In our cells, we have some very elaborate, and I'm not going to go through in detail, but what we could call molecular proof-checking machinery. So whenever our cells are replicating and the DNA is being replicated, there is machinery there to make sure that it's done correctly. Now that is a very, very accurate process, but occasionally the odd typo sneaks in. As any of us, when we're typing up our dissertations, will remember typos sneak in, even if you try your hardest. So there could be an error in DNA replication. There are also exogenous factors that can contribute to mutations occurring. So ionizing radiation, the exposure to radiation, increases the probability of a mutation occurring. So that they occur for a number of biological reasons. As an aside to our main theme and topic this evening, one interesting thought is that actually we need these faulty mutations to occur for evolution to work. So if you ascribe to evolutionary theory, mutations are the basis for what drives evolution, because you need a fortuitous positive benefit from a mutation to occur in order to develop gradual change within a species. So what I'm trying to say here is that evolution is the flip side of these here, or you might say genetic diseases could be considered the price that we have to pay for evolution, if we ascribe to evolution being the process that has been driving all of what's been going on. But that's an aside point. Now, what I'd like to do is just flip over, and well, that's the biology, but then consider a little bit around a biblical framework of how we can try and integrate the biology that we've been thinking about. The Bible starts in the beginning and claims that God created. And whether you read the creation accounts in Genesis literally or figuratively, that's not our debate this evening, but what they do acclaim is that God has a role as the ultimate creator. DNA and enzymes and metabolites are part of the created order. And in the beginning, God declared that it was very good. Part of why I was trying to baffle you with the biochemistry of the glycine cleavage system, as I said, was to show the exquisite complexity of biochemistry. Some people might go out on a dark night, stare up at the stars in the sky and be wowed by the creation. You can do the same looking down the microscope, looking at the complexity of what's going on in the cell and think, wow, this is amazing. This is pointing to some thought and design behind it. And one other aspect of the creation account that we don't always think about, but I just want to mention and we'll come on to again later, is the access at the beginning to what's described as the tree of life. Now the apple tree, or whichever fruit it was that Eve took the forbidden fruit from in the Garden of Eden, tends to get all the press. But actually, if you read Genesis, it's a tale of two trees. In Genesis 2, it says that the tree of life was in the midst of the garden and the tree of the knowledge of good and evil. And God famously forbade Adam and Eve from eating from the tree of the knowledge of good and evil, but they were at the beginning free to eat the tree of life. They had access to something that could sustain life in perpetuity. So in the beginning, it was all good. But then, as the famous story goes, the serpent persuaded Eve and Adam to disobey, to eat that forbidden fruit. And if we move on, thank you, Trevor, the account of the fall of mankind describes how mankind's rebellion resulted in a corruption of creation. There was a judgment on what had been made. Creation was broken, it stopped working properly. Childbirth became difficult and dangerous, and work would become toilsome. Man faced mortality, and by extrapolation, I think we can understand that normal DNA replication machinery stopped working perfectly, as it should have done, that deleterious mutations became a possibility, and so metabolic disease and other disease entered into creation. And as well, access to that tree of life was revoked. Can we just go back one step? Sorry, Trevor. So God said, lest he reach out his hand and take also of the tree of life and live forever. God stopped that access to the tree of life. And again, whether we read that figuratively or literally, there is something going on here, that God is putting a block on mankind obtaining everlasting life in his fallen and broken and diseased state. And that shows something of God's sovereignty and perhaps even his mercy in that account. So thank you. To summarise where we've got to so far. Are you following me so far? Are we okay? Why has this happened? And we have a biological model. We can explain why these diseases are occurring from a biological perspective. But we've also looked and thought a little bit about the theological model, thinking through the creation, the fall, how creation becomes corrupted, and as an explanation for why some of these things have occurred. There's a variation of the question as to why has this happened that we often get asked is from the parents, what did we do wrong? Have I done something wrong? Why has this happened? Have I done something wrong? Did I eat the wrong thing when I was pregnant? Did I do this? Did I do that? And it's a question that interestingly crops up in the gospel accounts as well. Jesus, if you remember the story in John's gospel, was confronted with a man who'd been born blind. And do you may remember the question that was asked, who had sinned that he was born blind? Was it him or was it his parents? Who had done something wrong? And Jesus' answer to start with is that it wasn't this man had sinned, it wasn't his parents had sinned. So while the biblical account holds that creation is broken and corrupt in general because of the sin of mankind, we can't take a specific incidence of an illness and say this has happened because of someone's specific action. DNA mutations occur because creation is corrupted in general, but it's not the fault specifically of the parents that they happen to carry these mutations. But the account of the man in John's Gospel continues to give some hints. Actually, there may be a higher purpose behind the suffering that he's facing. And Jesus said that in part his blindness was there that the works of God might be displayed in him. So there's something deeper going on, but it's not his fault directly. And as we try to help families come to terms with the diagnosis, I think we do need to help facilitate their development of their narrative, their understanding of what's going on, as to why this has happened, and to look beyond the biology. And sometimes they do reach out and are looking for answers elsewhere, and we can help point them in different directions. Okay, moving on then. We have a diagnosis. What can you do about it? Is there a treatment? And often for the rare metabolic diseases, we are faced with discussing being able to offer symptomatic treatments. So that's dealing with the problems that arise. So if there are seizures We can offer anticonvulsant medications. If the child is in pain, we can give them pain relief. If they've got problems with swallowing, we can give them a feeding tube. Sometimes there are some dietary measures that we can use. But ultimately, the treatment that we want for the child is something that is going to correct the underlying problem. We want something that can restore that missing function. And we're going to talk a bit around how we can consider the kind of medicine that we do in terms of a restoration or a restorative medicine framework. Can we replicate the function of the missing enzyme by doing something with their diet? Can we actually replace the missing enzyme? Or can we even, or should we even consider trying to correct the gene itself, the faulty gene at the very bottom of the problem? Now, before we go into look at that in a bit more detail, I want to just return to our meta-narrative, our biblical story arc, and see where we're up to. So after the first 3 chapters of Genesis, the rest of the Bible you could consider is a whole process of redemption, restoration, recreation, heading towards a final new creation. And it's interesting, if you look at the language that Luke uses in his gospel, and of course Luke famously was considered to be a physician himself, and he uses a lot of language that sounds like the sort of thing we do in medicine. So the hallmark of the Messiah figure is that the blind receive sight, the lame are walking, the lepers are cured, the deaf are hearing, and the dead are even raised. There's a lot of reversing of what has gone wrong with creation in what Jesus has been doing. And there are accounts where the disciples are told to go and do the same thing. So they're told to follow in his footsteps and to undertake this healing activity. So healing, curing, fixing what is broken is a good thing. And we are, in doing those activities and walking in those footsteps, reversing some of the effects of the fall and the corruption of creation, starting to undo what has gone on. Thank you if we move on. And there's also a symmetry, just to complete what we're talking about with the tree of life. If you remember, the account in Genesis has cut off access to the tree of life. But ultimately, the biblical story arc is heading towards the new creation. And the promises that you find in the eschatological accounts in Revelation, the end of time, is of the new creation, where it says, he will wipe away every tear from their eyes. Death shall be no more, neither shall there be mourning, nor crying, nor pain anymore, for the old order of things has passed away. And it's very interesting as well to see that in those accounts, looking forwards, that tree of life, that access to immortality is restored again. The leaves of the tree of life, which are on either side of the river, were for the healing of the nations. So that's really an interesting observation to where that whole account is heading. So by extrapolation, I think we do in medicine have a mandate to go and do some of these things, to take part in that restoration activity. We're given mastery over creation, we're told to go and subdue it, and so I think there is a restorative mandate, if we can put it like that. So what can we do with our restoration mandate? There are certain ways that we can do, and we are doing in the clinic, enzyme replacement therapies. So we, for example, can transplant a new liver in. If the enzyme system that we're dealing with, such as the urea cycle, is in the liver, you put a new liver in, you have replaced the missing enzyme and you've treated the condition. We can also use other things, and I'm going to talk a little bit more about enzyme replacement therapies and bone marrow transplants. We move on to the next slide. This tries to explain a bit about these. So for some conditions, such as those that are called the lysosomal storage disorders, the children are missing an enzyme that works to break down big molecules, and we can, in some instances, give them a drip, put an infusion up of a synthetic form of the missing enzyme, which then goes around the body and can treat the problem. We are correcting the underlying deficit. Now that doesn't get into the brain, it can't cross the blood-brain barrier and get in, so sometimes we use a bone marrow transplant, also called a stem cell transplant, where you take a donor's cells, you replace the patient's cells with those cells in the bone marrow, and then they can spread around the body, populate, and including in the brain, and they secrete the enzyme that's missing, and that can help to correct and treat the disorder. And we do that for Hurler syndrome, which is one of the commoner disorders that we deal with. So the next slide, if you're used to looking at Kaplan-Meier curves, this is a plot of how effective something is. Over the bottom on the x-axis is the age of the children that we're studying, and down the y-axis is the percentage survival. So over time, if you follow the red line to start with, that's what happens, the natural history of this disorder if you don't do anything about it. And you can see over time that the line goes down as fewer and fewer of the children are surviving. And by the time you get to 11 years or so, you can see that virtually none of the children would survive without treatment. With the yellow line, that's our enzyme replacement therapy. And you can see that it's an improved line. There's more survival. But actually the bone marrow transplant, the green line, not perfect, but we're seeing significant improvements over where we started. Good. So enzyme replacement therapies we can do, but then gene therapy and somatic genome editing, where we're actually correcting and treating at the level of the gene, is also a reality in the clinic. So there are a number of options. We can do what's called autologous lentiviral gene therapy stem cell transplant. It's really easy to say. So in this situation, you take the patient's own bone marrow cells, you gene edit them in the laboratory to put a corrected copy of the gene into those cells, then you give the cells back to the patient, and the cells then go around the body and replace that missing enzyme. And that's been successfully done in a number of patients. We have a couple in our clinic who've had this treatment for a very rare condition called metachromatic leukodystrophy. There are a number of other therapies that are in the research pipeline, but are very nearly there, using a number of gene therapy approaches for some of the conditions. So using different viral vectors or vehicles to get the gene into the body to correct the defect. And most recently, and again this is still in the research phase, but we're actively starting research trials of using in vivo genome editing, using techniques such as zinc finger nuclease techniques to insert a gene into a precise location in the liver cells, into a particular part of the liver cells, so that they can overexpress the missing enzyme. So it's kind of a brave new world. A lot of these technologies are starting to happen. And I would argue that at this level, where we're treating the patient in front of me, it is acceptable to be doing this genome editing because we're correcting, we're reversing something that is clearly broken, and it fits in within that restorative framework. There are questions around safety, and those need to be addressed, but I think in principle, if we can confirm safety of these techniques, then they are in theory, I would suggest, acceptable. There are a couple obviously caveats or limitations, and what I'm very much talking about is a restorative framework. Those of you who know the work of John Wyatt will know his classical description of a painter restoring an old painting. You're allowed to restore it back to where it was, but what you're not supposed to do when you're doing art restoration is add an extra pair of glasses or something to make it look better. You're meant to be sticking to restoring the original. And obviously, there is a risk that these same techniques could be used in what we could term enhancement. So if the Tour de France, at the moment, the dopers use steroids and other medications, could the future of doping being having some genome editing to make your muscles stronger and faster? Is that going to be a possibility? And I fear that may be the case. Now, that's not necessarily a reason not to do the techniques we've been talking about, but there are those obviously concerns. And there's also, I think, a real paradox, and this touches on events in the news at the moment, that there is a real paradox to how we value the lives of the children that we are caring for. The children are valuable. Life is valuable. There is a sanctity in their life because they're made in the image of God. And on the one hand, we spend a lot of our time doing all we can, despite the costs, financial costs for these precious lives, using resources recklessly. Some of these children are never going to live independently. They're never going to contribute to society in a financial sense. And yet we it is right that we spend, and we, in some people's minds, waste resources on these children just because they are made in the image of God. But sometimes we do have to acknowledge that death has won, that there is a futility in some of the treatments, and the burden of treatment outweighs anything going forwards. This phrase, whispering hope, is one that I've borrowed from the mother of one of the patients that I looked after for a number of years, including through his final days. And his mum talks about whispering hope, that sometimes death isn't the worst thing. And if we are ascribing to this meta-narrative, actually, death sometimes isn't the worst thing, and we can offer that hope. So while we hold a high value on the lives of the children that we care for, sometimes we have to acknowledge that our treatments, however cutting edge, haven't worked, and that death is going to win. How are we doing for time? Good. Okay, finally, the third question I want to move on to is just to consider what are the implications for future children. We've said where ignorance is bliss, 'tis folly to be wise. So said Thomas Gray. Well, sometimes in the clinic and in the information that we've found out, we are wise to facts that have massive implications, and we have to deal with it. And in particular, when we're explaining to parents about these recessive metabolic conditions, we have to discuss what's termed the recurrence risk. And if you remember the model that we had of our genetic disorder, both parents are carriers, and every time they have another child, there is a risk that the next child could also be affected. And out of those 4 options, there's one out of 4 has the condition. So every time they have another child, there's a 1 in 4, 25% chance that another child would be also affected. And for some of the conditions that we'll touch on, if we have time briefly, the mitochondrial conditions, that risk can be even higher. So that then leads the parents to face a number of options or choices. And what are the choices that are available to parents if they're considering having more children? Now, some parents may have got to the point of having all the children they were going to have. Maybe they choose not to have more children. But if they do wish to have further children, many will choose to run the risk, or take their chances, or leave it in God's hands. It's very interesting that the language that they use when you're talking to them is very insightful as to where their thinking is coming from. But actually also the language of the person who is counselling the family is also very insightful, and we'll think about that in a moment when we consider how can we counsel them appropriately. So in the context that they do decide to just have a spontaneous pregnancy and take the risk, run the risk, whatever we're going to call it, in that situation we would offer to test the baby once the baby's been born, see if they're affected or not. And oftentimes, particularly for the conditions where the babies become very unwell very quickly, we step in and start treating them from the moment they're born to try and prevent them becoming unwell. And there are some situations where if we know about the risk of it, we can actually completely stop the disease from causing problems. Other options, other parents may opt not to have their own biological children. So they may choose to go down an adoption route, or they may choose some form of donor gamete IVF, so test tube baby, where there's a donor sperm or egg that's provided to try and avert the risk of the condition being passed on. But even in that situation, that's not a guarantee that you won't have some other condition. And we've got patients in our clinic where, tragically, the child is affected with a condition because the donor also was a carrier for whatever condition. And we'll consider again in a little bit some of the other concepts around parenthood if we think about the mitochondrial 3-parent babies in a moment. Other parents will opt for antenatal testing. So often they'll have an amniocentesis at a relatively early stage of pregnancy to see if the child's affected. And if the child is affected often, although not always, they would then choose to terminate the pregnancy and not go further with it. Another option is that of pre-implantation genetic diagnosis. So here it's a form of IVF again, so test tube babies. You create a number of embryos, you then test them to see if they're affected with the condition, and then you only select and implant unaffected embryos So you only choose the embryos that are not affected. Those that are affected by the condition are then left and succumb. Now obviously that also generates lots of other issues. You may make more embryos than you need, and some of them may be healthy, and that is a conundrum in itself. What should you do with the spare embryos? And finally, we're not there yet, but embryonic genome editing is very much on the horizon, and that's using genome editing techniques to correct the genetic defect either in the gametes or in the very early embryo, effectively treating the disease before it's even had a chance to have any effect. And we'll have a few words about that in a moment. Now there are, as you can imagine, a whole raft of dilemmas that come out from the fact that we have choices. Firstly, and these are questions, not statements, do the parents have a moral obligation to avoid having another affected child? Is it a moral obligation to avoid a child having a condition and so suffering from it? Do they have an obligation to avoid the impact of having another affected child on their wider family and the costs to society? We can probably agree that it would be better for the child I have in clinic, the existing child, not to have a given condition. It's probably better that they don't have it. Can we agree that it's better not to exist in the first place than to exist and have the condition? If the only way that you can avoid suffering is by ensuring that that child doesn't exist in the first place, is that the morally right thing to do? And then we have to ask, is there a sliding scale of badness as to when this might be the situation? A very, very minor condition, I don't know, an abnormal hair colour, you would consider as a very minor thing. It's not going to have a big impact. But at what point on the sliding scale of conditions is something bad enough to say this is an intolerable condition that the child shouldn't be allowed to have? And then if there's a treatment, does that change the balance of the equation? And what about when a new treatment becomes available? And if we look a few years ahead, and if we speculate that embryonic genome editing will be possible, does that then obligate the parents to utilise that to avoid having another child with a specific disorder? I haven't got answers to all of these, but there are huge questions that these are opening up. Thank you. Now, I wanted to say just a word to non-directive counselling. This is the concept that when we're presenting these choices to families, that it should be non-directive, that our own beliefs shouldn't be prejudicing how we present the options. And it is true that we should, in our clinical practice, for those who are in clinical practice, be careful in how we are presenting the different choices and ensuring that we are fair-handed in how we're doing that. But yet often families are not after non-directive counselling. The question, what would you do in the situation, is one that we often get asked. And obviously I can't answer that question. I've not been in that situation to know what I would do. But I think it is fair to say that actually the default position of many professionals is towards antenatal testing and potentially abortion being the right thing to do. And I think there is a degree to which the counselling that is offered isn't non-directive across the board. I think there is much more that we can be done to ensure that that full range of options is made available and explained. And certainly one of the aims of my practice is to make the care and treatments that I provide to my patients so good that the parents might risk doing it again with another child. And can we do that? So then just a few words on germline gene editing. This is the whole concept of doing embryonic gene editing, editing things right back at the beginning. Now this is different to the genome editing trials and work that we're doing in the clinic at the moment, because that's treating a patient and it will not go any further than that patient. But if you edit errors in the genome right back at the beginning in the embryo, that is then going to be passed on from generation to generation. If that child has children, that genome editing, the edited version, is what's going to be passed on. And there are a number of issues around that. Is that a right thing in itself, to be editing the whole human genome? How do we prove safety? We can't prove it's safe now unless it's tried. And what's a very interesting thought, and I don't have an answer to this, is how on earth do we consider the consent process? Those of us who are in paediatrics are quite used to taking proxy consent for whatever procedure we're doing from the parents because the child's too young to do it. But here we are doing something that's going to have an impact on many future generations who won't have consented to having had their genomes edited. Now, I think in theory, maybe you could argue, if you go back to our restorative principle versus enhancement, if we could generate just one embryo, if we could treat it, implant it, and not avoid all the issues of other spare embryos, would that be acceptable if we are editing and taking out what is obviously a very deleterious mutation, even if that is then going to be passed on and that corrected version is going to be passed on down the line? That's an open question in my mind. I think the whole caveat again around enhancement is, however, an issue. And if we were to start doing this and generating a subspecies who have got superhuman powers because of our genome editing, that obviously is a very different ballpark. And I think also there is a really fundamental issue to remember, that behind a lot of these techniques, even if we could say in principle this is acceptable as an end, the means by which we get there has been an awful lot of research on human embryos. And it's a classical question: do the ends justify the means? Is a good outcome a healthy child? Does that endorse the process of what's gone before? And I would argue that perhaps there is an argument to say that actually the means themselves we need to be considering, is it ethical as in how we're getting there? And the status of the embryo, and we don't have time to talk about this in great detail, but I think is of paramount importance. And if we have a high view of the status of the embryo, if we subscribe to the view that conception is when a human is created and that they bear the image of God, then that is going to impact to a large degree on all of these considerations. What is the status of an embryo? Is it a future person? Is it a presently existing individual who bears God's image? Is it just a symbol of human life? These are all theoretical concepts that are put around embryos. But we can perhaps talk about that more afterwards. Thank you, Trevor. I want to just say in the last few minutes, just a few words, as it's a very topical subject and one that's widely debated. The whole question around three-parent babies. And I'm sure you've seen in the news, and some of you may be working in the kind of the field of ethics around this. Mitochondrial disease in some instances is different to what we've been talking about so far. We've talked about recessive conditions, but for the mitochondria, which are the powerhouses of our cells, the batteries of our cells, they are made up of proteins that come both from our main DNA, from the main genes, but also the mitochondria house a small amount of their own DNA, the mitochondrial DNA. And that is only passed on from the mother, because the mitochondria, when a baby is formed, are only passed on from the egg, from the oocyte, into the baby that's created. And if a mother carries mutations in one of the mitochondrial genes in that mitochondrial DNA, there is a risk that to a larger or lesser degree the baby that's formed will inherit a bad dose of those mitochondria and suffer with some very significant disorders, because if your batteries are not working, you can imagine that's going to cause havoc with your body. Thank you. And there are these techniques where that are being developed, and the Newcastle group have developed and are at the forefront of that internationally, of developing approaches where mitochondria can be provided from a third party in that process of creating the baby. And there are different approaches to this. So the top one is maternal spindle transfer. And so this is done prior to fertilization. The nucleus from a donor egg is removed and replaced with the nucleus from the mother's egg, and then it's fertilized with the dad's sperm, and the combination of the nuclear genes comes together, but with the donor's healthy mitochondria. There's another form, pronuclear transfer, where the switch is done after fertilisation, and that requires a donor embryo, which is then used to house the combination of the parent's DNA. So here we've got a very complicated technology, and in the UK, thank you Trevor, the legislation has moved forward. We are still waiting for the first child to be produced from this process, but there are a number of issues around this. Obviously there's embryo research involved again, and there's a question as to whether this mitochondrial transfer technique, is it analogous to an organ transplant? Are we just doing what we do in clinical practice? We replace a liver if it doesn't work. Here we're replacing some mitochondria. Or is it more like germline genome editing? And those mitochondria are going to be passed on down the generation, so it's not just something like a liver transplant that sits with a patient. It is going to move on. So there is that analogy with germline genome editing, except you are just donating mitochondria. There are lots of issues around the safety and whether the technique would actually work. And to come back to our concepts of parenthood, this really challenges, and this is perhaps what has grabbed the attention of the public, is that here you have a child being formed with 3 biological parents. Now, we all have 2 biological parents, but actually our genome is a combination of our 4 grandparents. So there is perhaps some scope and looking to see how that would fit together. And then there are those non-biological aspects of parenthood, adoption being the paramount example. I again don't have a final view on whether that in itself is acceptable or not, but I think it's all of the issues around this are enough to give us pause for thought. And I would direct you, if you haven't read it, to a very good journal that Trevor has is the editor of, that has actually got some very insightful papers, both from the Newcastle group and also from others to counteract that. So just some final thoughts then in the last couple of minutes to draw this all together. If we go back firstly to our biblical meta-narrative, thank you, next slide. We've been drawing on this idea that creation fallen and creation being corrupted is why things are as they are. We don't have time to fully expand on this now, but part of the meta-narrative of the Bible states that corruption of creation is judicial. It's a result of God's judgment on something that has gone very wrong. And our ultimate illness is what we could call sin sickness. It's not our metabolic disorder. And that's only dealt with in the biblical account through the redemptive work that was done at the cross, by which God graciously provides a gateway into the new creation and access again to the tree of life. And there's a lot more that we could talk around that, but suffice it to say that even the most advanced of genome editing techniques is not going to effect that ultimate cure that we need in that framework. That requires a much greater physician. So, Trevor, we've explored these 3 questions that have come up in the clinic. Why has this happened? We've thought around the biological model. We've thought around a theological framework. We've asked, what can we do about it for my child? And I've tried to explain how I practice and how I think in terms of this restorative model, the restoration mandate that we have in our practice, and even pushing the envelope out to consider genome editing. Can that fit in with that? And I think in some contexts it can. And we've asked, what are the implications for future children? And we see some of the very painful and the difficult choices that are presented to parents. And I think our view of the status of the embryo is going to determine to a large degree how we respond to those questions and how we go about our non-directive counselling. We don't really have time to think about this in detail, but how can all of this inform our decision-making at the personal and clinical level? Having a fluency in the language of meta-narratives, having an idea actually that people are getting, there's more to life than just the biology, is going to be important in helping people cope with the diagnosis and in making those important treatment decisions and those end-of-life decisions. And trying to think creatively in our counselling and how we present those options to families. And at the wider policy level, as I said, advocating for restorative medicine, and in some instances pushing out what is the current science and taking it to where we can, but at the same time restraining some aspects of those advances where we don't think it fits in with what is a sound approach. And ultimately we are trying to safeguard the most vulnerable, those who don't have a voice. And finally, the frailty that we see in those most severely affected of children often serves as what you could call a memento mori, a reminder of the mortality that we all face, that we're all broken, some more visibly than others, and all of us alike need a much bigger cure than genome editing is ever going to be able to do. So I hope that's been an interesting, stimulating, hopefully, idea and a tour through metabolic diseases, and thank you very much for your attention.